Hello students, now we are going to discuss about the types of plant tissue culture, right? So when we talk about the types, there are different kinds of plant tissue culture. There is a slightly confusion that whenever it will be asked in the exam, students they will write the methods of plant tissue culture, right? So that's what there is a difference between the types of plant tissue culture and methods of plant tissue culture, right? So whenever this question will come into the exam, we have to read the question carefully whether it has been asked the type or methods, right? So generally the types of plant tissue culture, it is there in the syllabus, clearly it is mentioned. So that's what there are different types, means where we are using the different part of the plant and we are cultivating into the different nutrient medium, then we are getting the any new plant, that is called the types, right? What are the different types? Maybe shoot, root, leaf, etc., right? So let's try to understand what are the different types of plant tissue culture. So the first one, it is the root tip culture, right? So when we talk about the root tip, as the name indicating, root tip we are going to use like tip of the root, right? Tip of the root, it is sterilized, right? So surface sterilization we have studied, right? So by using up the different types of sterilizers, sanitizers, maybe sodium hypochlorite, bromine water, something like that we have to use, and we have to sterilize, then transferred into the medium, suitable nutrient medium. Obviously, just like a microbiological culture, we need to have here again the medium. That medium will have the all the nutrient which is required for the plant cell to grow a new plant, right? Then that will that nutrient medium, it will grow, continue the growing, and it will grow and provide the several roots. From one tip of the root we are going to get the several roots. After 7 days, the experimental culture we are going to establish, and this root material actually they multiplied in the continuous culture, right? So then so on, then those roots can be used for the generation of the new plants, and it can be planted into the different places, right? So where we are using the examples are very very important. In so many books the examples are not available. These I have collected from the different research article, right? So that is very important. You have to give this one example: root tips of the peas, corn and cotton can be the cultivation can be done by the root tip culture. The second one is the leaf culture, right? So when we talk about the leaf, right, so different leaves are here the minimum requirement is 800 micrometer leaf it is required, and again we are going to do the surface sterilization. They sterilized by different the sanitizers, maybe sodium hypochlorite, I told you already, right? Bromine water, hydrogen peroxide, etc., depend on the which part we are using. So here we are using the leaf. So solidified medium we are going to place them, and where they remain in healthy condition and they will grow slowly. The cell division, cell multiplication, it is going to taken place. Okay, this culture we are using for the life plant, right? Miracle plants, miracle leaves can be used, then begonia, etc., are the example for the leaf culture, right? Now the third one which we are going to mention here is the shoot tip culture, right? So when we talk about the shoot tip culture, we are going to use the 100 to 1000 micrometer shoot, that is the size, minimum size which is required to grow the new plant. Then again we are going to have the sterilization, and we are going to put them into the medium where the plant growth regulator also we can add. This we are doing in the case of corn, peas, in the sugar solution, etc., that can be done. The fourth one, it is the flower culture. So as the name indicate, flowers we are going to culture, and we are going to get the plants 2 days after pollination. What is the exact time of the collection of the or excise of the flower? We have to wait we have to find out the pollination period or the time when pollination has been completed or it had been done. After 2 days, that excise the flowers, and again we are going to have we are going to sterilize that one, then sterilize, then immersion into the 5% of calcium hypochlorite, right? That we are doing to then again we are going to wash with the distilled water and place into the culture tubes, agar medium, that is there will be in that culture tube there will be the agar medium and the plant growth regulator simultaneously. So this culture we are using for the rose flower, orchid flowers, tulip and the lilies flowers, and there are so many other flowers can be used for this particular culture. The fifth one, it is the anther and pollen culture, right? So when we talk about the anther and pollen, as the name indicate, anther is going to excised and into the, after that anther or pollen grain, we have to, which that part it is going to use, and that has to be placed into the nutrient medium. The immature anther and pollen usually grow abnormally, right? So that we have to take care. We have to, whenever we are selecting the anther and pollen, that has to be matured one. These things we are doing in the case of tobacco or in the case of datura plant or datura cultivation, right? In those anther and pollen, we are going to use this particular culture. The sixth and important, that is the ovule and embryo culture, right? So here what we are doing is embryo or is dissected from the ovule, right? So what we are doing here, embryo is dissected from the ovule, right? So here you can see here that ovule and put into the culture medium. Again we are going to have the culture medium there only. So this particular types of culture, it is having two types, protoplast culture and hairy root culture, right? So the first one is the protoplast culture, right? So what are the protoplast? Naked cells of varied origin without having cell wall. Generally plant cell will be having cell wall. So the protoplast means it is the naked cell from the varied origin. It may be root, stem, fruit, flower, bark, any location, but it will not be having the cell wall, that we are going to rupture, right? So by the different method, like for example here protoplast, it is isolated and centrifugation, it will be subjected for centrifugation at a specific rpm for the certain period of time. Then the cell wall will be broken out, and again that we are going to put into the nutrient media. We are going to seal that. If it is, if you are taking the petri plates, that has to be sealed properly, right, by the wax or something like that. Here the protoplast is going to be have. Then it has to be inverted position. We have to keep in the BOD incubator for 2 to 3 weeks, right? So biological oxygen demand, that is the special incubator. It is not the normal bacteriological incubator, it is the biological oxygen demand incubator, where oxygen supply it is going to be there, right? So that's 2 to 3 week we are going to have. This thing we are doing in the case of potato and in the case of tobacco cultivation. So let's try to have the brief on the protoplast culture. So when we talk about the protoplast culture, here is a certain types, right? Like whatever the leaf we are going to select, that has to be sterilized properly, right? Then epidermis has to be removed from upper epidermis. If you are taking that, after removing the epidermis, we have to put them into the centrifuge, right? Centrifugation. And once you will put the centrifugation, the cell wall it is going to break down, and the cell will is going to expose. Those are called a protoplast. That protoplast we have to plant. The planting of protoplast we are going to do here, right? Planting of protoplast. Then when you will plant with the suitable nutrient medium and plant growth regulators, they will start division. The first division it is going to take place like this. The protoplast cell it is going to divide just like a mitosis meiosis, right? In that case, right? So in this then again we are going to have the colony formation. When we are after first division, if it will be taken into the nutrient medium, proper suitable nutrient medium, the colony formation will start. The protoplast it started dividing. Then it is going to form the callus. In the same way, after few days, after further more days in the incubation period, right? Then this callus differentiation we are going to observe in the same conical flask. Then it is going to generate the plantlets in a such a way, right? So then these plantlets ultimately it is going to give you the young plants that can be planted into the proper pot or maybe transplanted into the open field, right? So it is the step by step procedure of the protoplast fusion, that you can see that leaf sterilization, epidermis removing, then centrifuge the protoplast, the planting of the protoplast, first division of the protoplast, then we can see the colony formation, then we can see the callus, then we can see the callus differentiation, then regenerated plantlets, and ultimately young plant we are going to observe for this case, right? So then the another one it is there, the hairy root culture, right? So in hairy root culture, the second type of oval and embryo culture. So in this large number of small fine hairy roots has to be excised or collected and into the culture medium. And in this we are doing the vinca and vinca, rose and sugarcane can be done, right? So these are the different methods of the plant tissue, different types of the plant tissue culture, which is very important, and it will be asked for the 5 marks 10 marks question, right? So at least you should know the names at least, right? Root, leaf, shoot, flower, anther, pollen, oval, embryo, protoplast and hairy roots. Thank you.